topic that on gene transfer is part of that idea where microbes are evolving, their genomes are evolving through these processes of transformation. In this case, that's where we left off. By the way, I'm hoping to get my laptop squared away and all the necessary programs loaded for us to try iClicker on Friday. All right, so if you could come prepared on Friday to do that. And when I meet with the CTL people, Tom, I'll ask why you're ending up in the other class that I'm teaching. I have no idea. I asked my other class, I said, hey, do you see, BI 303? They're like, no. So you were all special. Apparently you can be in two places at once, be in both of my classes. So technology, I tell you, sometimes makes me scratch my head. Okay, we left off here looking at transformation, the uptake of naked DNA, which it's usually small bits of DNA or sections of DNA that gets taken up. This is the gram-positive process. I looked through your 6th edition. They omitted this topic in chapter 9. It used to be in it, and they took it out. And I'm like, what? So sometimes gram-positive bacteria get short-tripped. I'm guessing that one of the authors, there's a new author. I'm wondering if he's a medical microbiologist, and medical microbiologists tend to be a little bit more partial to gram-negatives because gram-negatives are the ones that cause so much serious disease in humans and other animals. So anyways, I still want you to know this though, okay? So this is the idea of gram-positive type bacteria have this mechanism of sensing their environment through quorum sensing. That's how they kind of tally up who's around them, how many of B subtilis cells are around themselves, or maybe some other type of microbes. And then if the conditions are right, they will go ahead and do this small signal cascade, which results in the building of that transformosome, which are a bunch of proteins that get put together in a complex in order to allow for kind of like a channel to form and allow for that DNA to pass through. And so when they've studied microbes in the lab and they're looking at, how does this transformation process come about, this is what they're seeing with all the gram-positives that they're studying. The gram-negatives, on the other hand, when they look at those in the lab and look to see how do they take up naked DNA, it's a little different. They don't see those competence factors showing up. So basically that's saying that there's no quorum sensing going on with gram-negatives. I think time will tell on this idea. I do think that quorum sensing does make sense to go ahead and do during this process of transformation. So I would imagine in the near future this may change a little bit. But for right now, they're saying, yeah, we don't see any quorum sensing. And so what they're saying is, well, either they're always competent, or if you starve them in the lab, if you manipulate their conditions where you purposely starve them, then you can get them to do this. And so you have to appreciate that a lot of these studies are done in an artificial setting, in a lab, in a tube, very artificial. And so a lot of this information is based on that. And, like they're saying, oh, there's no quorum sensing. Well, I think you need to kind of wait and see and do more studies and see what happens. They have some anyways. Some gram-negative microbes have this special pilus, which is, it kind of looks, well, it's not a flagella, but it extends off the cell, and it's protein based. And there's different kind of, the plural of pilus is pili, P-I-L-I. There's different kinds of pili out there. And they give numbers, but this particular pilus, when they've looked at some of the bacteria, they actually use a pilus, like the tip of the pilus will like latch on to DNA and kind of drag it into the cell. And so there's some interesting mechanisms that some of these microbes have evolved in order to get access to this naked DNA. So when you're going to learn pretty quickly here that gram-negative bacteria have a special outer membrane, and they do see that there's some issues that outer membrane causes some extra challenges. And so we'll get into what that is. But basically, the big picture idea here is that they use a special pilus in order to drag in the DNA. Here's an image of that, trying to show this long, well, they don't really show the pilus length of it, but they're showing here how the pilus has latched onto it and is kind of like retracting and bringing in the DNA. So, you know, when they starve the cells, so what they do is they pop them into broth, but they don't give them many nutrients in that broth. And so they trigger a starvation environment. Kind of makes sense that they might hunt them for some DNA to deal with that. You know, is there some other genes out there that could help me under this critical situation? Microbes, by the way, have several strategies to deal with starvation and other, or like drought, you know, going through dryness. There's many things that they can do, and we'll keep talking about that especially as we get into cell structure and function. Okay, so in my mind, I think of transformation as kind of the easiest method. It's pretty straightforward in the sense that they need to get access to bits of naked DNA, and they find ways to take that in directly. So here's another idea. So this is the second big one, conjugation. I always think about conjugating verbs in my different foreign language classes that I took. But basically what we're looking at here are two cells in this image, and you see that yellow thing in between them. So this is the idea of, again, using a special pilus, and that's the yellow thing that's stretching between the two microbes. So when they watch conjugation happen in the lab, they say cell-to-cell contact is necessary. That pilus is needed to touch both we call it a donor cell and a receiver cell. And so they actually refer to it as a sex pilus, because remember that these ideas of horizontal gene transfer is the microbial version of having sex. And so sometimes it's nicknamed the sex pilus. And so what they see happening is the pilus will be attached to one of the cells that made it. Okay, so they make the pilus and they get it all set up, and then that pilus is kind of searching for another nearby cell to latch onto. And then once it makes physical contact with another cell, if everything's a go, they'll kick into the conjugation process. And what they actually see, and they've taken some good video of this, where that pilus will make physical contact with the second cell, and then the pilus retracts again, almost like it's like, you know, yanking that cell closer to itself. And you can actually see that under the video during this process. So conjugation involves plasmids being transferred from a donor to a receiver. And remember now, plasmids are usually circular pieces of DNA. They're separate from the chromosome, much smaller than the chromosome. And they contain genes that generally aren't needed on a minute-to-minute basis. They're kind of things that are needed occasionally. And so plasmids can get transferred through conjugation methods. What they're showing here are a donor and a receiver cell or recipient, and they've color-coded the plasmid kind of pink color here and the chromosome green. So what they're showing here is that the donor's on the bottom here, and so they're showing that the pilus has made contact, it's retracted, so they're physically much closer together, and then they build kind of like this tunnel system between them, and they transfer part of the plasmid. Now, if you are the donor, you need to pause and think about this for a minute. All right, so plasmids are double-stranded pieces of DNA that they've just been circularized, okay? So if these plasmids can have genes on them that are useful, if you're a donor, would you want to give away your whole plasmid? No. Evolutionarily, you know, trying to survive, giving up your whole plasmid, that's a little too generous. You don't want to be that generous. So what they do is they give away one strand of the plasmid. Remember, it's double-stranded. So what they can do is just donate off one strand, keep the second strand with themselves. Now that makes sense because that is allowing a recipient cell to gain access to these genes, but the donor retains that as well. And then once the donation of that strand is done, each cell will then make the complementary copy to bring it back up into the double-stranded state, and then it's functional again. Okay, so these plasmids have all the necessary genes on them for the pilus formation, to do the conjugation event. So they're kind of special in that regard. And most of this work has been done on E. coli. By the way, Escherichia coli is like one of the guinea pig, like probably the guinea pig microbe out there. When I was studying for my, when you go for a PhD, you have to do qualifying exams. So usually your second year into your program, you have to go through qualifiers where you do a written test and oral exam. And when I went, you have your committee, and I went to each committee member and I said, what do you want me to know? And they would tell me what I should be studying, what I should study. And one of my mentors, I said, well, what do you want me to know? And he gave me like 3 volumes of E. coli research, like they're in book form because it's so well studied. He's like, here, know this. Gave me 3 huge volumes. And I just kind of looked at him. It was a joke, but seriously, he didn't want me to know a lot of that. What was in those volumes, because E. coli, it's just such a workhorse guinea pig. And we know more about E. coli than probably any other bacteria out there. And so E. coli is gram-negative. You're going to work a lot with E. coli as well. So E. coli has this plasmid, the fertility factor plasmid. I don't really care that you know that so much, but you may see in the chapter readings, they talk about the F factor. And then they say, well, the donor cell that has the plasmid, we'll call it F plus, and then the recipient will be F minus. So that's where that's coming from. It's from the so-called fertility factor plasmid. So what's going on? Here is kind of a step-by-step, but I'm not going to get into the details. One of the reasons I show this image is just to get you to appreciate that it's more complicated than what I'm saying, and that it is quite a remarkable, quite a remarkable event. And so here you can see the sex pilus was built by the donor cell on the left. It extends out, latched onto the recipient, and then you can see in the next image down how the pilus has retracted and has pulled that recipient cell so that they can make physical contact with each other. So that's what we mean by, there's got to be some good physical contact, because then you got to get like a tunnel built. And so they're showing the process of the tunnel essentially being built. And then here you can see the one strand of the plasmid getting transferred over. That gray there, that's just one strand of the plasmid getting transferred over to the recipient. And then when that has occurred, now you can see that each cell, the donor and the recipient, are making a complementary copy. So by the time this is all done and they physically separated, the donor still has an active plasmid, that's good, and then the recipient has gained a plasmid. So you don't need to know all the details, like in the yellow box, all the words here. I'm not going to get into, the ORET and all of the 5' end and blah, blah, blah. All right. So and then looking here, you can see like the building of the tunnel and getting everything going is complicated. That's what I want you to know. It's complicated. And then verbally, what we just went through is what I'm looking for, that you can describe the process like what we just went through. Questions on the conjugation idea? Sometimes that's not the end of the story. sometimes you have, let's say the recipient cell gets that plasmid. Usually plasmids stay separate from the chromosome, but not always. So sometimes we see that that plasmid actually pastes into the chromosome of the recipient cell. And if that happens, you'll see they'll then start calling it the HFR, high frequency of recombination. Recombination is referring to the idea that it went into the chromosome. It recombined into the chromosome. So sometimes that can happen. Not all the time, but sometimes. And so why that's interesting is that if you imagine that plasmid going into the chromosome, then you need to appreciate that under the right conditions, that plasmid will kind of cut out of the chromosome at some point. That tends to happen. And you can manipulate that in the lab. You can force the situation. So that's what they do when they study this. But so it will go into the chromosome, and then under the right conditions, the plasmid comes out. And when it gets cut out of the chromosome and becomes separate again, sometimes it will take flanking chunks of the DNA chromosome with it. And so that's kind of an extra way for these plasmids to have really nice special genes that could get transferred now to some other organ, some other cell. And so you can see how you might end up with some interesting plasmids that can have these extra. So now in the lab, they've done some really careful studies where they can get like huge sections of the DNA or huge sections of the chromosome to transfer and get like become part of the plasmid essentially. And then they make another conjugation happen. And they've even seen like an entire chromosome. But you got to remember, this is in the lab. All right. In the real world, I don't think a microbe is going to give up its whole chromosome. So this is under a very artificial situation, but they can get it to happen. And so it's pointing to the idea that getting chunks of the chromosome into the plasmid and sent off to a recipient cell is probably happening in real conditions, in natural conditions. So here you're seeing this idea in visually, they're trying to show the plasmid. The circular thing on the top image here, and trying to show that it's getting incorporated into the chromosome. And again, the details of this is not needed. This isn't a molecular biology class, but just this idea that you can get that plasmid to integrate into the chromosome. And then at some point, it's going to cut out from the chromosome. Fancy word for that is excise. And I cut it out essentially of the chromosome, and it can take some genes of the chromosome with it. And so it's got some extra genes that it didn't have before it integrated into the chromosome. And so that's another way for now, if you imagine this plasmid getting transferred to another recipient cell, it's got some extra genes there on the plasmid that recipient cell could definitely take advantage of. So they often refer to this as F prime plasmid when you get the plasmid now containing some chromosome genes. So again, I'm not too concerned that you remember exactly what F prime means. I'm more concerned that you understand the overall process. Like, can you recognize that or describe the idea that sometimes the plasmid gets into a chromosome, cuts out of the chromosome, and takes some chromosome DNA with it? I'd rather you know the concept than the term. Okay. So a visual of that, they're trying to show that as the plasmid is cutting out, excising out, it's taking some flanking, typically some flanking DNA with it. So conjugation, they first studied this in E. coli, and they could show 2 E. coli cells doing it. And that was a big deal. I mean, to show horizontal gene transfer, it's like, wow. Then they started playing around, and more scientists got on board, and they're like, well, who else can do this? Is it just E. coli? And they started to find lots of other microbes can do this. And then they stumbled on this one, which is amazing. where you have microbial plasmids being transferred into plant chromosomes. I mean, that was just mind-blowing when they found this one out. So this is the idea of crown gall disease. This is how the pathogen causes crown gall disease. I'm sure if you've been hiking around in the woods, which I hope you do on a regular basis, go out and enjoy the woods, you might have seen these types of growth, particularly on tree trunks. And these growths, they kind of look like a tumor, right? In the botanical world, we don't really say tumor, we say gall. But that means basically it has that, that's what we refer to. Tumor, which is growth, is a gall. Crown is just referring to the region of the stem that you see this showing up. So what this is all about, and this species I do want you to know, okay? I want you to be able to remember that crown gall disease is caused by Agrobacterium tumefaciens, because this again is such an important microbe. If you go at all and do any kind of work where you have to transform a plant cell, like introduce new DNA into a plant cell, you're likely to use Agrobacterium to get this to happen. So let me explain it a little bit. So this is an interesting pathogen. It causes crown gall disease, which normally, by the way, doesn't kill the plant. Have you seen crown galls, these tumors on, and the plants overall in the, in the middle of the summer, they look fine. They got green leaves and they're doing okay. So normally this disease doesn't kill the plant. It might weaken them a little bit. So if another pathogen comes in, there might be a little bit more susceptible to the, but there's a really good one on Bluff Street. There's a house on Bluff Street with a tree, a huge gall, and they tend to decorate it like around Christmas time. I think it's Santa. They make it a Santa face, and then Halloween they do. I got to go and knock on their door and say, "Can I take a picture of this?" Because I would like to put it into this slide. But anyways, so sometimes they have some really interesting designs to decorate. This pathogen, this bacteria, has a special plastic that's been called the TI plastic. I stands for, well, TI stands for tumor inducing. So this is a plasmid that has the genes on it to cause this tumor looking growth. And the bacteria infect the plant. So if you can imagine like a leaf tree, where the bacteria, like the rod-shaped bacteria in this lower image, it will get attached to the tree trunk cell, and then it will get in. And then what it does is it will integrate its plasmid into the plant chromosome. And on that plasmid are genes for telling the plant, grow, 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 grow. In this spot where I live, I want you to grow. And so that tumor or the gall, and if you think about it, that's pretty nice for the bacteria because they live in that, right? So it's like it's telling the bacteria, like, make me a home, grow. And so you get these cells growing, and if you take a chunk of that and look at it like you see it's chock full of the Agrobacterium. So that's pretty slick. Then there's also extra genes on that plasmid that the plant will express. And one of them is a nitrogen-containing compound. It codes for the production of a nitrogen-containing compound. And what's really slick about that is it's a nitrogen-containing compound that the plant makes now because the plant is expressing that plant DNA. But the plant can't even use that nitrogen-containing compound. So the bacteria say, make me a home and go through and make that nitrogen-containing compound for me that I can eat. And so the pathogen has a pretty slick process in place. It gets a home and food just by transferring the plasmid into a plant chromosome and getting the plant to express and do all the work. And so when they found out that this Agrobacterium is doing this, again, they were just like, wow, because now they're like, hey, could we take Agrobacterium and do this like with plants we want to introduce genes into? Because if you've taken, well, I'll just tell you flat out, getting genes into plants, it's really hard. It's very challenging to do that experimentally. And I won't go into the details right now, but it's a challenge. And so when they found out how easily Agrobacterium does this, they got super excited because they're like, hey, we could use Agrobacterium. We could put our genes that we want in its plasmid and then make, introduce the bacteria to the plant we want to get this gene. And so they've been doing that over and over. The nice thing about Agrobacterium is it's got a pretty broad host range, like it can infect a lot of different plants. And so that's nice from an experimental perspective. And so a lot of plants have been transformed this way. We get our genes of interest, like from drought tolerance or to fight off certain pathogens, heavy metal exposure, and things like that. You can use Agrobacterium. So it has revolutionized. Am I saying it right, mate? It's been revolutionary. How about that? To see how using this method can really advance the plant genetics. And so it's been really, really, really useful in the botany world. This table is looking at some of the genes, the phenotypes that will result if those genes on those plants get expressed. So here you're seeing on the right column all the different microbes that are producing these, that have these plasmids, okay? And you can see that it varies. Lots of different species are being listed there. And then you see over and over, wide range, wide range. And then if you look on the left, you can see some pretty concerning things like, where is it? Antibiotic resistance. Okay, antibiotic resistance. So a lot of the microbes that are antibiotic resistant, they're getting that resistance through conjugation processes. And we're finding out that many, many wide-range bacteria are capable of doing this. So that's why medical microbiologists are so concerned about antibiotic resistance, because it can spread like wildfire from one microbe to the next using this plasmid transfer conjugation. And so it's something that isn't going away, and they're trying to figure out ways to deal with them. But on the other hand, there are some other things on here that can be very useful. So, for example, degradation of oxygen or naturally herbicides. So now we're thinking about the idea of remediating contaminated areas. So, for example, soil that gets contaminated by these different industrial chemicals, you can use microbes to deal with them. So, and that's bioremediation. That's what we're talking about. So you use microbes to clean up the pollutants because they have the plasmid that can allow them to actually use those pollutants as nutrition and food. And they break it down to harmless stuff and clean up the environment. And so that has been something that has been going on, that, they've been using microbes for many decades now for remediation. So there's many different traits or phenotypes that you can see on here. Some good, some not so good. So basically what we're saying is that when a microbe obtains these plasmids, it can definitely allow them to do a new behavior and to adapt to an environment that maybe before that plasmid, they were struggling to survive. And so it gives them a selective advantage in their habitat. And you can imagine that, if you have a pathogen in you, and you go to the doctor and he says, yep, you're sick with a bacterial pathogen, I'm going to give you an antibiotic. Then you start taking that antibiotic, if there's some microbe, one of the microbes in you has this plasmid that can allow for resistance, you could see how doing conjugation in that host, those microbes, if they could pull that off, it could allow them to survive your antibiotic treatment. And so, of course, they're looking at that very carefully. But basically, they even say, because it's so efficient, this plasmid transfer, they often use the word infectious. It's that easy for microbes to transfer plasmids, and that's what's got them so concerned, because that allows the trait or the plasmid with those genes to get spread rapidly in in populations. And now we know, especially with that agrobacterium study, now we know that you can have this happening between really unrelated microbes and even microbes and other organisms in different phylum and different domains. And so certainly the more they study, the more they're finding this to be true. More and more examples. This is not from your book, but again, this image is trying to get across the idea of this. It doesn't have to be like E. coli to E. coli. You can get a lot of different combinations of who is sharing these plasmids. So you can see, domain archaea to domain bacteria and vice versa. Then you see bacteria to the eukarya domain. And this is not a complete list, eukarya to eukarya. So often it's a fungus. Fungus are eukaryotic microbes. Okay, so this is something that is, the more they study, the more it seems to be like, oh yeah, they can do it too, And so this is something that is an ongoing research to look at this, especially when it comes to antibiotic resistance. That's a big one that they're really studying carefully. Okay, so conjugation, plasmid transfer. Those genes on the plasmid can confer very advantageous traits. Here's another idea. Transduction. There's two flavors of transduction. One is called generalized. Okay, so as you're studying this, it's a really, really good idea to compare and contrast. What does generalized transduction mean? And then the other one we'll talk about is specialized. So I want you to know the differences. What's the difference between generalized versus specialized? What's similar? Okay, and try to keep them separate in your head. Don't confuse the two. Okay, I'm saying that now because I've seen in the past, examples of students that don't study that carefully enough. They start to confuse the two. Okay, so generalized transduction. Okay, so now this is probably the most complicated version of gene transfer because we have a middle entity involved, which is a virus. Okay. So now we're seeing it's going to be ending up to be a bacteria to bacteria transfer that we're going to look at with the help of a virus. Okay. So here you're looking at a cell on the top image here that is getting infected by a virus. So you see, for some reason, my laser pointer is not working. There's the virus. All right, so the virus contains DNA. The virus is going to inject its DNA into the bacteria. By the way, phage, the word phage, that's short for bacteriophage. Those are viruses that only infect bacteria. They don't infect humans. So bacteriophage, and then they shorten it down to phage. Some people say phage. I say phage. All right, so that's the purple stuff coming into the cell. Then you see the host DNA in gray. Okay, what's happening to the host DNA in the middle cell? What's going on there? It's getting chopped up. All right, so this is one of the reasons we call it generalized transduction, because when the viral infection kicks in, it triggers the host DNA to fragment, to get chopped up. So now in that image, you're seeing the DNA that the virus injected, plus the host chromosome DNA. And what they're showing in the bottom image, what they're trying to show, is that some of the viral particles, instead of all being purple, like what you started with, You see some are purple and some are gray. What they're trying to show there is that some of those viral particles picked up the fragmented host DNA, and it's totally random. That's why we say it's generalized. It's just random bits of host DNA floating around, and then the viral particle captures, just happens to capture this piece or that piece of the host DNA. And they pack it into their, we call it the head region of the virus. Okay, so generalized is referring to this idea that the host DNA gets fragmented, those fragments are floating around, and any old random piece is going to get incorporated into the viral particle. Okay, so that's not the end of the story. This is just showing the first portion of it. Okay, so now that those viral particles, there are some that have the original viral DNA, the purple stuff, but then there's a bunch of viral particles that have the host DNA. So now imagine they're all ready to go. That cell is going to lyse or break open. Those particles spew out. So that's what they're showing here. And now those particles can go on to, those virus particles can go on to infect the nearby cell. So here you see in this case, the viral particle that has a lot of host DNA from the first host is getting attached to another bacterial cell. And now we'll inject that DNA. So effectively, what we see is that bacterial DNA is getting transferred from the first host to the second host, but it's through the help of that virus. Does that make sense? Okay. And they say it's generalized because it was almost bits and pieces of the host DNA just randomly floating around that get randomly incorporated into a virus. So now the first host, it's kind of a bummer because the first one is dead. It got lysed over. But the second host could benefit because if this virus particle injects host DNA from the first host, well, that's good. It could confer an advantage. Plus, what often happens is this virus is incapable of causing this cell to lyse. It's lost the ability. It's lost too many of its own genes to do that. And so this second host may survive it. Not always, but it may survive. So now this second host, let's say it survives it, plus it has new genes that it's never had before from the first host bacteria. So the second host could win big on this. First one, not so much. That one's dead, but the second one. So you are getting DNA transferred from the first host to the second, just through the help of that virus. Okay, so that's generalized transduction. Questions on generalized? Let me compare that to specialized. Okay. So let me just kind of come in here. That's as far as it lets me zoom, hey? All right. So at the top, let's look at that. So you can see different colors. Looks like the host chromosome is light green, and it's the pink one that they made for galactose utilization. And then the middle stretch there is dark green, and that's so-called phage DNA. So that means they're not showing the virus affecting the cell. They've skipped that part. So what they're showing is viral DNA got into that bacteria, and is it floating freely? No. Okay, it got pasted into the chromosome. That's a huge difference. So compare and contrast. This is the contrast. We see in specialized transduction, the viral phage, bacteriophage, that's the virus, its DNA got put into the host chromosome. That's a huge difference. Okay. Then they're saying two things could happen once that occurs. Let's go, well. All right, so let me just briefly mention the normal. You see, eventually what can happen is that phage DNA, under the right conditions, stimulus, that phage DNA will cut out from the host chromosome. And it cuts out totally clean, where you're just back to the bacterial phage, viral DNA, all intact. But for specialized transduction, I want you to look at the rare event. Okay, so with the rare event, what happens is that somehow that cell gets stimulated or gets exposed to some kind of situation where that viral DNA will get cut out. But instead of cutting out cleanly, it takes flanking DNA with it. So that's what they're trying to show. It's going to take some of that lactose and show that. And so basically, the flanking DNA, the left and to the right of where it pasted in, that flanking DNA can come with it, and it's a sloppy cutout instead of a precise. We're only going to cut out the phage DNA. Now it's phage DNA and flanking host chromosome. So that's what they're trying to show here in the bottom right. You can see that pink showing up in the as part of the viral DNA. Now, let's see if I can. So now you can see all that viral DNA is not just viral DNA anymore. Now they're showing it in packaged up, and then this host cell is bursting open to release those viral particles. And some might have just the viral DNA, but some will have viral DNA plus host chromosome. So now you have to imagine some of those viruses going to a second host. And now when they inject their DNA, they're injecting some viral DNA, but a lot of host chromosome DNA too. And again, it might happen where that second host will survive that because the virus, the genes that allow the virus to like totally take control and kill off the cell eventually won't be there. Some of the host chromosome is taking care of it. So you have a situation where that second host may survive this. And if it does, maybe some of those host chromosomes in the first host that the virus attacked, maybe some of those genes are useful for that second one. So they call it specialized because it's special regions of the host DNA that's getting packaged, not just random stuff floating around. It's the flanking DNA of where the phage was pasted in. Okay, the flanking DNA. Questions on the specialized transduction? Let me go back one. Find where the word is. I think it's here somewhere. Oh, there it is. Okay, this word. Lysogenize cell. That, anytime you see that, if someone like microbiologist was working with you in the lab and they're like, oh, these are lysogenized cells. Here are some lysogenized cells. What they mean is they know that these cells were infected with phage and that some of the phage DNA pasted into the chromosome. They know that so then we say that cell has been lysogenized. I'd like you to know that term, please. This is a different image, kind of side by side, compare and contrast of generalized versus specialized. That's from your book, but I thought for study purposes it might be useful doing the comparison of the two. So, you know, this is a virus that's involved in this process. And of course, for most host cells, getting infected by a virus is like, not always such a good thing, right? So in this particular case, this can be kind of a danger idea. Yeah. 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 Yeah, usually if you have a normal event, then what happens is all these stages spew out, that one's toast. They go to nearby cells and do the whole thing all over again. And again, they'll break open the cell. We'll talk for sure in this class about viruses and how they do their thing later in the semester. So bacteria, when they get attacked by viruses, and I'll mention this again when we cover viruses more detail, they do have ways to try to fend them off. So it's not like they're totally at the mercy of viral particles. And so if you've taken intro to cell and molecular, you might have worked with restriction endonucleases in the lab. You can thank bacteria for that. That's what they have. That allows them to try to cut up foreign DNA. So when the virus injects its DNA, they have these restriction enzymes. And if they can cut the DNA into pieces, it becomes ineffective. The phage is like, can't do anything. And so it's like their own immune system in a way, really, if you think about it. And so all of those restriction endonucleases that you work with in that class and any other like cell biology or advanced cell, that's you can thank the bacteria for that. They're doing that. They can also protect their DNA. So we see, do I have that picture? Oh yeah, the next picture. They can protect their DNA by methylating. Those orange balls are methyl groups that are put onto the DNA, and that also can protect their own DNA while they chop up the virus. Okay, so it's a way for them to just make sure that the viral DNA is getting chopped up. Okay, we got to stop there. We're out of time. We'll finish up this idea of gene transfer on Friday, and then we'll get into cell structure and function, and we'll try the iClicker, hopefully, for the first time on